Welcome to Chapter 14, Non-Adaptive Evolution and Speciation. So after today's lecture, you should be able to list the three causes of non-adaptive evolution and explain how they are different from natural selection, as well as describe how genetic drift and gene flow affect population diversity, and predict how genetic drift and gene flow contribute to the survival of a species living in an environment that is rapidly changing. Last but not least, you will be able to define speciation and explain how it can occur. So these are all non-adaptive forms of evolution, which what we've been talking about already has been adaptive evolution or natural selection. So to begin talking about non-adaptive evolution and kind of what your book talks mostly about is something called population genetics. So what exactly is population genetics? Population genetics is basically just the study of genetic makeups of populations and how genetic composition changes. So it's understanding how a group of organisms is coping with environmental changes. So with population genetics, you're studying quite a few things, but mostly relying on DNA and looking at how DNA is changing across a population over time. So is that population evolving? How is it evolving? In which ways is it evolving? Okay, so when we're looking at population genetics, we're looking at an entire population, right? And last class we learned that populations evolve, but individuals do not. So we're looking at an entire population and we're mostly concerned with their genetics. So when we're looking at a population's genetics, we're looking at their gene pool. A gene pool is a total collection of alleles in a population. So you can see here, if we're looking at our little mice over on the left-hand side, if we're looking at this, each mouse here has 2 alleles for each gene. It can get a little bit more complicated than that, but for this class, for these purposes, assume that each gene just has 2 alleles, right? So you're getting one from mom, one from dad. That's always the case, but we're just looking at one gene in particular right now. So that's really what I want you to focus on. Don't get too caught up in that there's lots and lots of genes that these mice actually have. Just focus on the one gene with 2 alleles. So since each mouse actually has one gene and 2 alleles that we're looking at, you can see the different alleles that they have. So we have red, green, as well as blue, and you can see that they have the allele frequencies down below. So we've got 6 out of the 30 alleles are blue, 12 out of the 30 are red, 12 out of the 30 are green. Keep in mind, there are 30 alleles. So how many mice is that for? 15, right? So 15 mice each have 2 alleles. You have a total of 30 alleles in that gene pool. So population genetics is basically looking at what that gene pool looks like, let's say now, and then going back in several generations and looking for a change in the allele frequency or looking to see if the alleles have changed in the number that they have. So you can look and see after a few generations, right? We're looking now, and now instead of having 15 mice, how many do we have? We have 19. So now with our 19 mice, we have 38 total alleles in this gene pool, and you can see that our frequencies have changed a little bit. So our blue has increased, now we're at 0.32 or 32%, red has decreased slightly, and green has also increased. So it's kind of interesting to be able to look at these allele frequencies changing over time to study whether a population is evolving or not evolving. So by doing this again, we're just identifying the allele frequency. Kind of a refresher from the last course, but any change in allele frequency over time would lead to a population evolving. And what we learned about last time was natural selection, right? So those are good consequences usually, and it results in the population becoming more adapted to its environment, and that's a result of natural selection happening. Sometimes you can have bad or neutral consequences, but for the most part, you're becoming more adapted to your environment. So you know that those things are going to make you as an individual more fit. And the higher fitness you have, the higher likelihood you have of passing on those alleles to the next generation. Okay, so we have natural selection happening, but what other forms of evolution are there? We've heard of evolution and we often associate it with natural selection, and usually that is something that's happening, right? eventually over time you have to become more adapted to your environment. Otherwise, the likelihood of you surviving and having a high fitness is probably relatively small. So you have to keep evolving. So what is actually causing the changes in these allele frequencies? One way, like we've talked about, is natural selection, right? And through natural selection, our population is becoming better adapted. So this is what we call adaptive evolution. In case you don't remember, this entire lecture is mostly about non-adaptive evolution. So when we're looking at non-adaptive evolution, it's forms of evolution that are still happening, but it's not necessarily making the population better adapted. And this is something that we study pretty heavily with population genetics, and we look at the DNA pretty frequently and almost solely when you're looking at non-adaptive evolution. With natural selection, keep in mind you do look quite often at the DNA because it's not always a trait or a phenotype that you can easily observe or see in an individual. But with non-adaptive evolution, you have to be looking at DNA because you don't necessarily see a lot of mutations or genetic drift or gene flow, which we'll talk about in just a second. But non-adaptive evolution, the forms of it can either be mutation, genetic drift, or gene flow. Okay, so first up we have genetic drift. Genetic drift is a change in allele frequencies between generations that occurs purely by chance. So, just like it says, this is purely by chance, this is completely random, genetic drift is. So say you have a subset of a population that reproduces. If that subset of the population reproduces, that subset of alleles is represented in the next generation. So if we have a mom and dad, let's say, that are going to be reproducing, the only alleles that are going to be passed on are of those that reproduce. Everyone else that has possibly lower fitness is not going to reproduce. They're not going to pass on their generation or their alleles to the next generation. Overall, genetic drift, like I said, is a chance, but what it does, it's still evolving. It's still a form of non-adaptive evolution, but it overall decreases the genetic diversity of a population. This is possibly a little bit difficult to understand at times because it doesn't seem like something that we, it's a little bit weird to think, okay, we're decreasing genetic diversity but still evolving. And what this means basically is that we don't have as many alleles represented in the population because by chance or by genetic drift, you aren't passing all of them on to the next. This is one example of genetic drift that's actually in your book, and you can see that this is, again, just one example, and this is if the populations get separated. So you can look at the big red dots that are there, and they're showing 15 study sites that this guy named Munchie South studied in New York City. So the different shades of green represent percent canopy cover, and you can see that urbanization has fragmented green spaces into separate, very distinct patches. So urbanization just meaning all of those big city skyscrapers and buildings that have gone up in New York have effectively cut down the landscape, right? So you have lots of different populations being separated, possibly lots of animals being killed as well, but you're definitely separating them by these urban buildings. So some of these patches you can see are connected by greenways and some are not, but what you find is that you have different populations that have different alleles in them. So when you're looking at this, if you look at this whole bar right here, I'm trying to figure out a way to actually highlight it for you guys so you can see. It's not working for me. Here we go. Okay. So we're looking here, and when we look here, what we can see is that each single little bar is actually the bar of one mouse's genetics, right? So we're looking at their entire genetic code there. And you can see that they're kind of grouped together, and they're actually grouped together. They have really similar colors based on the areas that they're from. So depending on where they're from, which population they're in, they actually share very similar DNA to the others. And you can see there's still some variety in there, but overall there's not that much genetic diversity. You can still tell that they have very similar genes, very similar alleles for those genes within the one population. And this is probably because they're not able to get outside of their population, so they're probably breeding quite often in this population, and possibly because they're well adapted to that population in that environment, that they're actually having higher fitness and able to pass on those alleles. So again, this is purely random. The animals don't necessarily have a choice in what sort of urban buildings are going up, or say that we're tearing down a forest, whatever it might be. The animals don't necessarily have a choice in it. It might also be the result of some natural disaster or some natural event, even like a flood or some sort of dam that builds a new river or anything to that effect. Anything that can separate these populations by chance can potentially lead to genetic drift, which would potentially lead to decreasing the genetic diversity of that population. One other form of genetic drift is called a bottleneck effect. So a bottleneck effect is just kind of like what we were just talking about. So it usually happens with rapid habitat loss, although there are other ways that it can happen too, such as natural disasters. One example of a bottleneck effect is actually with the cheetahs, which we'll talk about in just a second. But when you're looking at our diverse original population over here, we have, you can see the blue, red, and green alleles. So we have all of those, and we have them in different frequencies, right? And because we're great population geneticists, we go through, we look at the DNA of this population, and then we look at it after some event, right? So a bottleneck event would be like something like rapid habitat loss, which would be us going through and urbanizing an environment, building a skyscraper, possibly building a freeway, anything to that effect. And after that, you're obviously going to be separating certain populations. You might even be killing off some individuals in that population. So when you go over after the bottleneck, now we're seeing a less diverse bottleneck population. So you can see which alleles have we completely eliminated. We've completely eliminated the red alleles, right? They no longer exist. All we have are blue and green, and they're in very different frequencies than they were before. And you can see this population after several generations, you're obviously increasing the frequencies of these because you no longer have the red, but whatever one is more fit in this population might also continue to increase, possibly knocking out another one. So we're really leading to less and less genetic diversity here. A very similar bottleneck event was with our cheetahs. Ignore how terrible my writing is, but with cheetahs, I think it was about 10,000 years ago, we had a big ice age that essentially knocked out most of them. And cheetahs are really interesting because they're so genetically similar that if you were to take a cheetah today and give them a skin graft, they can actually accept it from any cheetah. They have no immunorejection just because they're that similar. So that is a result of a bottleneck effect, which is categorized under genetic drift. Okay, so again, decreasing our genetic diversity. Whew, this is bad writing, but that's okay. You guys will get over it. Okay, so that is one example of non-adaptive evolution. Another example, I don't think I have here in the slides, but another example of genetic drift is what's called the founder effect. Sorry guys, this pen is not great. It's good for highlighting, but not for writing. So a founder effect is kind of just like it sounds. So it's also completely by chance usually, but say you have a few individuals that leave a population and go and found some other area, right? So they're moving to a new area. The only alleles that you were going to see in that area afterwards are from these founders, right? So this is what we call a founder effect, is a few individuals moving from one area to another, and we're also still decreasing our genetic diversity because the only alleles that can be passed on in this new population are those from the founders that came before. So again, a couple different examples of genetic drift. You have founder effect and bottleneck event. Okay, so we keep talking about genetic diversity, but why is genetic diversity important? So there's a few different reasons. One is that a diverse gene pool gives the population more flexibility to survive in a changing environment. This is something that's really, really important, especially today or in the future when we're facing pretty severe climate change. And there's other examples as well, right? The whole world is constantly evolving and our populations are evolving with it and adapting to it as much as they can. Sometimes it's really difficult to adapt to some extreme event really quickly, but the more alleles you have in a population, the better chance you have to be able to survive that. So say you have extra alleles for surviving in really, really cold places or really, really warm places. Having different alleles in a population basically allows you to select for those and ensure that your population can survive and eventually evolve and adapt over time. So the more genetically diverse the population is, the more ways it has to adapt. Oops, sorry, didn't mean to go. Can I go back? Okay, well, I can't go back. But that's essentially why we have genetic diversity and why it's so important is we want as many ways to adapt as we possibly can. So while we're adapting, we are often losing genetic diversity, but it doesn't mean that we lose it completely, right? Just because you are not as well suited for your environment as maybe some other individuals are, doesn't mean that you might be killed off right away. It might mean that your alleles become less and less frequent over time. Maybe there won't be as many of you, but with your chance of being there is also an increased chance of adapting to a change in that environment. So it's in the population's best interest to have as much genetic diversity as possible. So we went over genetic drift and how that can decrease genetic diversity. So how could we potentially increase genetic diversity? So in order to increase genetic diversity, it's pretty simple, right? We have to bring in new alleles. How do we bring in new alleles? That's where mutation comes in. And again, mutation is completely by chance, so this is still a non-adaptive evolution. The other way that we can bring in new alleles and introduce more genetic diversity is by something called gene flow. Gene flow is literally movement of alleles from one population to another. And we'll go over both of these in a little bit more detail right now. Okay, so with gene flow, right? So we have a population and usually gene flow is just moving, again, we're moving from one population to another. So what we're getting here is we're getting our alleles moving over and about, and it's through migration and interbreeding. So you guys will hear interbreeding a little bit. You will also hear about inbreeding here in just a second. Interbreeding and inbreeding are very different things, so make sure that you understand the terminology there and what each of them mean. I'm sure you've heard of inbreeding before, and we'll talk about it more extensively here in a second. But with interbreeding, it's just any members of a species that can breed together. So you have the same species here, right? We have mice over here, but they're in different populations. So these populations can interbreed with each other. They can interbreed within the same population as well. That's interbreeding. So the effect that they have on gene flow is you have migration as well as interbreeding. So you might have a red mouse that's going over from this population over here, and it's traveling over to this population. And you can see now that before gene flow, we only had the red and green. Now, all of a sudden, after our gene flow has taken place, we had the red one come over, and then we started possibly reproducing after a few more generations. But immediately after gene flow, you can see now we've got 2 new red alleles. So that changes our percentage to about 6%, where it was at 0% before. So we are increasing, and similarly over here, we have no blue. So we're actually getting a little blue guy coming over. And then with our green, we're just increasing our likelihood of having green alleles since we mostly have these little red guys. So, like we were talking about earlier with urbanization, that can oftentimes prevent gene flow, right? So that's what we are talking about a little bit with genetic drift as well. With urbanization, if we're preventing gene flow, it's also leading to a lack of genetic diversity, as well as potentially inbreeding. So urbanization, if we're putting up a new building, if we're putting up a freeway, I don't know if you guys have seen those little wildlife bridges, but say you've got a freeway that you put up in the middle of a big old forest and you have all these trees, right? And now they're building these little cool wildlife bridges that go over, and it's like basically all green. So they have these cool wildlife bridges. It's hard to draw on here. That like would go over, say, the freeway, and then they can actually, animals can move back and forth. It's a really cool idea. And it sounds really nice in theory, but how much the animals are actually able to use this and learn this probably has yet to really be tested. So that'll be kind of interesting, but it's at least a nice thought for trying to help them and trying to increase a little bit of genetic diversity instead of just screwing everything up by urbanization. But now we get into inbreeding. Okay, so we're going into inbreeding now, and this is again causing a lack of genetic diversity. It's lowering genetic diversity, and it happens when we prevent gene flow. So when we happen to prevent individuals in a population from moving to a different place. So inbreeding is probably just like you've heard about before. It's just mating between closely related individuals. Okay, it doesn't change the allele frequency within a population. If you have mom and then you have dad and they're mating together, I know dad would be a Y, but I'm just putting examples. If you have them mating together, the only possible alleles we can have are these guys, right? So if they keep mating and then their kids mate, then you just keep getting the same alleles being passed down, and it really doesn't change. That has a few different problems associated with it, but one of them is it increases the proportion of homozygous individuals to heterozygotes, right? Because we're passing down the same alleles, so the odds of having 1 and 1 are now pretty slim. Now the odds of having homozygotes instead are becoming greater and greater. This can lead to something called inbreeding depression. So this is when your closely related individuals are more likely to share the same alleles, right? Because they came from the same, basically same mom and dad, right? And then mom and dad have a couple kids, and then let's say that these kids mate. They're essentially going to have very similar, if not the exact same alleles. So when we cross these guys, we're again getting more of the same alleles. So if they're more likely to share the same alleles, then we have some negative reproductive consequences that are coming into this population. Then becomes associated with a really high frequency of homozygous individuals, but mostly our main concern is homozygous individuals that possess harmful recessive alleles. So this is like some of the recessive diseases that we learned about. Those are really common in some inbreeding circles, depending on which organism you're looking at. But it's kind of interesting. I don't know if you guys have heard of the blue people of Kentucky. You can Google it right now. It's pretty weird and interesting, and I think some of it might be a bit exaggerated. But when you're looking at it, these people actually have a little bit of a blue pigment in their skin, and it's because they're very closely inbred. So you had that blue pigment show up in some gene somewhere, and that allele just kept being passed down through this family and through these generations because they kept inbreeding. Okay. Another example I think your book talks about is in the Amish community having polydactyly. So polydactyly means that you have an extra finger or toe. And they had someone, an immigrant from Europe, I believe, I can't remember where, maybe Germany, came over and was part of this Amish community. And essentially, again, through inbreeding, they had this inbreeding depression. And this polydactyly, this recessive linked trait, is now being passed down to many in the Amish community. So the Amish community actually has a pretty high level of polydactyly in it, which is kind of interesting. Okay, so that's inbreeding. Remember, inbreeding is different than interbreeding. We'll talk a little bit more about interbreeding here in a second. More about urbanization. So these guys that are in your chapter that are studying these mice in New York, it might seem a little bit weird, but it's a pretty cool place to be able to study evolution, right? Because you have such high impacts of urbanization. So what they did too was they looked at the difference between country mice and urban mice. So they're the same species, but they wanted to see how different some of their genes might be. So they specifically looked at genes that would be associated with immunity and response to stress. And they realized that the country mice weren't actually as well adapted for these as the city mice were. So the city mice actually shared more alleles of these genes that were associated with response to stress. So they had a little bit less allele diversity in it. And it's probably because those have been selected for again and again to help them cope with the stresses of city living. So having to cope with, an influx of humans, lots of different species, lots of different things around them, lots of noise, as well as helping them cope with immunity, right? So they're probably in a more toxic environment in the city than they are in the country. They have a lot more toxic chemicals, a lot more pollutants, all of that good stuff. Well, not really good stuff, all of that bad stuff. So our country mice here are definitely not as well adapted for the city living as these guys are. And it's because these alleles have been selected for and they've been passed down from generation to generation. Okay, so this is a super important table for you. Memorize this. It will be on the exam in one form or another. So again, we've gone over natural selection, and that is pretty much our only form of adaptive evolution. Everything else is non-adaptive. Okay, so it's completely random. It's not necessarily because it's favored in any way, but again, an important thing to know is how genetic diversity is is affected. Usually in natural selection, it does decrease, right? Because we're still selecting for the alleles that are more favored in our environment. So we are decreasing our genetic diversity, but oftentimes not quite as much as we would be with, let's say, genetic drift. Again, just because these are happening doesn't mean that they're mutually exclusive, and it doesn't mean that they always decrease genetic diversity, although that tends to be the way that they're happening. Mutation and gene flow are both actually increasing our genetic diversity. Okay. And remember, it's important to remember evolution is pretty much happening all of the time, and all 4 of these forms of evolution are happening at the same time. So we don't just have mutation or genetic drift happening. They're all happening at the same time, and really we can't stop a lot of them from happening. So they're going to happen whether we want them to or not. All right, so here's where I'm going to lose you guys and where we're going to start getting into a little bit of math. So, as population geneticists, when you're studying populations, it's really, really difficult to actually find out if they're evolving, right? And it depends on which population you're looking at. If you're looking at bacteria, it's really easy to see if they're evolving over short time spans because they're evolving fairly quickly because they can multiply over generations and generations within an hour. But if you're looking at something that's longer lived, like almost all individual organisms that we probably think of, or even humans, that's really hard for an individual to see in their lifetime, right? Because you're looking at different generations, and it's also really hard to get a good representative sample of a population because sometimes there are just so many individuals. So what they do is they actually use this Hardy-Weinberg equilibrium. There's an equation for it, which you'll see here in just a second, to get a baseline for populations. So Hardy-Weinberg equilibrium is pretty much impossible to have in nature, which we'll go over in just a second. But it's basically the idea that in a non-evolving population, again, very hard to have, in a non-evolving population, allele and genotype frequencies do not change over time, right? That makes sense because we know that evolution means that our allele frequencies are changing over time. So the opposite should be true as well, okay? So when we're in Hardy-Weinberg equilibrium, we can use this to identify genes that have changed because of evolutionary mechanisms. So population geneticists will use this as a baseline to judge if a population is evolving. You guys ready for the equation? Okay, this is the equation. Learn it, memorize it, love it. So the equation is p squared plus 2pq plus q squared equals 1. Also important to remember that p plus q, equals 1. Okay, so p is, well, here, p squared is the frequency of homozygous dominance. So that means p are our dominants, okay? So what would q then be? Q is our recessives. I'm sorry, guys. I got to get a better pen, but okay. I don't know if that's any better. So P squared is the frequency of our homozygous dominance. So 2PQ is, you guessed it, that's our frequency of heterozygote. Okay, so we have 1P and 1Q. Okay, so we've got one homozygous and one dominant that's leading to our heterozygote. And then Q squared is our homozygous recessives. So the total proportion of all of these has to equal 1 or 100%, right? Same exact thing. Oops. So that's why we can also use P plus Q equals 1. For Hardy-Weinberg equilibrium to exist, there must be 5 necessary conditions. Again, this is essentially impossible in the natural world. So the first condition is that we cannot have any mutation introducing new alleles into the population. Again, this is pretty much impossible because we have no say over mutation. Our DNA regularly mutates, and so do other organisms' DNA. So if it mutates and it's not caught in some checkpoint, then you're going to have a mutation. Second is that we have no natural selection favoring some alleles over others. So we're not becoming better adapted to the environment we live in. We also would have to have an infinitely large population size. The debate over infinity is too vast for this class, but an infinitely large population size is still not really plausible. If you had an infinitely large population size, you would therefore not have any genetic drift. So this is, again, something that we do have, but in our equilibrium world, we do not. We also would have no gene flow, so we wouldn't have any new influx of alleles from neighboring populations, no migrating, no interbreeding, and we would definitely have random mating of individuals. So this is the one thing in the real world that can happen and does happen quite frequently. Okay, so here is a little bit of a figure to hopefully help you out with that equation, right? So we've got the equation down here, P squared plus 2PQ plus Q squared equals 1. And you can see we've got mom over here, dad over here, right? So both mom and dad have one dominant and one recessive allele, right? So you can see our dominant in the red and our recessive in the green. So mom and dad are going to mate and they're going to produce offspring. And we are looking at the likelihood of which kind of offspring they will have. So we have P crossing with P. So we have P squared. We then have P crossing with Q. So we have PQ. We happen to have 2 PQs, which is why you're seeing 2 PQ here. Then lastly, we have Q. Oops, sorry guys. And then we have Q again, so we're getting Q squared. Okay, so that's where the equation originally comes from. It's not too difficult of an equation, but you do need to remember it, and it helps. Again, it's basically to give us a baseline to judge whether evolution is occurring or not. So we can take this and say this is what we would expect if the population is not evolving. So if we get something different than that, then we know the population is evolving. And what we do is we plug in the frequencies of different alleles here. So remember when you're doing this, if you know you have, let's say, 6 out of the 10 are red and 7 out of the 10, oh my gosh, what am I doing? 3 out of the 10, ignore that. 3 out of the 10 are green. So these are our frequencies of just our recessive and our dominant alleles, right? So if we're looking at the dominant, our red, our P, we know that we have 6 out of 10. So when you're going to plug this into the equation down here, you don't put 6. Instead, you would put the frequency. So 6 out of 10 equals 60%. Oh my gosh, this is so bad. 60%, which equals 0.6, right? So 0.6 is actually what you would plug in to P. Okay, so P squared, oops, would be 0.6 squared. And again, all of this has to add up to 1. So if you know one of these, you can find out the others. Okay. So you will have a class activity on trying to figure out a little bit more with Hardy-Weinberg equilibrium, and hopefully it'll make a little bit more sense. But we're going to continue on with the chapter and get into what a species is and how they can speciate. Okay. So what is a species? What makes a species different from another? Because we look at, let's say, ants, right? And I don't know about you, but ants pretty much all look the same to me, although you guys will be testing this out in lab a little bit later this semester, and you will see that not all ants are the same. And I'm sure we know that, right? We know that not all ants are the same, but when we're looking at them or when we're smashing them because they're annoying us or we have an infestation, they all pretty much seem the same, right? So what makes them a separate species? They can't just look morphologically different, right? They can't have different body parts or something like that. It's got to be a little bit more to it. So when we're defining a species, we use what's called the biological species concept. Biological species concept is basically just the idea that a population of individuals whose members can interbreed, so this is important, they can interbreed, not inbreed, interbreed, and produce fertile offspring. Okay, so these guys have to be able to do both of these things. Otherwise, it is not a species. Different species cannot mate for several different reasons sometimes, but it all boils down to the fact that they are reproductively isolated. We'll go over some examples of what reproductive isolation means and how it can happen to different species or different populations even. But remember, in order to be considered a species, you have to be able to interbreed and you have to produce fertile offspring. So sometimes species or individuals can interbreed, but they do not lead to fertile offspring. That is not considered a species. Okay, so reproductive isolation. Reproductive isolation is just the idea that basically that you cannot mate with another species because of some isolation, right? So there's a few different types of isolation. One is an ecological isolation. So this is when species are just in completely different environments. So as an example, you have an Arctic fox and a desert fox, and they're in just such different places, they're really never going to encounter each other. Could they mate otherwise? Possibly, right? But it's a moot point because they're never going to see each other. So this is ecological isolation. There's also temporal isolation. So the example here, we have a leopard frog and a bullfrog. And these guys mate at very different times in the year. So one is in early spring and the other in early summer. So because that's the time that they're mating and they're trying to attract their mates, they're never going to line up on these cycles, so they will never actually mate with each other. That's temporal isolation. So temporal meaning time. We also have behavioral isolation. So they might have different mating activities or be attracted to sort of different mating rituals. So this is an example with a prairie chicken here who is not attracted to the mating display of our ring-necked pheasant. So these two would never ever meet because they are not attracted to one another. That's our behavioral isolation. We also have mechanical isolation. So this one's probably fairly obvious, but if the mating organs are incompatible, they're not going to be able to mate. And a very classic example here is of different pollinators that you might have. So bees are usually designed depending on which kind of bee, but bees usually are pretty generalist in how they feed on pollen. We learned a little bit about bees earlier this semester. But these guys here, these hummingbirds, they really like these flowers that they can dig into with their beak, and they're really well made to be able to pollinate these flowers, versus our bees tend to go for these more broad, flattened flowers. So they're not pollinated by the same sorts of species, so they're mechanically isolated. We also have gametic isolation. So remember, gametes are your sex cells. So if your gametes cannot unite, if you cannot take, say, a dog sperm and a cat egg to form a zygote, which would then develop into a fetus, then you're not going to be able to reproduce. We also have hybrid inviability. So this is when two different species can mate, so say a goat and a sheep, but the zygote they form does not survive. This one I'm a little bit unsure of for this example. This is a real thing, hybrid inviability, but do me a favor and look up something called geep. You will not regret it. They are absolutely adorable, and they do survive, and I think that they might actually fall more into this next category, which is hybrid infertility. So with hybrid infertility, you have viable hybrid offspring. So say these guys mate, or here we have a zebroid, right? So a zebroid is the product of a zebra and a horse. So they do survive, they do become a zygote and eventually become a fetus and end up living, but they cannot produce offspring of their own. So again, they don't qualify as a species because they don't meet this second requirement. Okay, and I think that geep actually fall into the same category, but hybrid inviability is a thing, and I can't think of a good example of it right now. But yeah, look up geep. You'll be so happy you did. It'll take out the bad memories you have from the blue people of Kentucky. Okay, so when we're looking at species, especially here in Hawaii, we have a really unique situation because we're really, really far away from anywhere else in the world. We're the most isolated place on the planet, I think. So that's really special, and that's something that's kind of interesting because we have a lot of species here that look very similar to other species in the world, but they are actually different, right? And it's because of this reproductive isolation. So this is where we get speciation. So speciation is essentially developing or becoming a new species. So it's the genetic divergence of populations leading over time to this reproductive isolation and then forming a new species. Okay, so that's what speciation is. A really classic example is with Darwin's finches. You can see the Galapagos Islands here off the coast of South America, off Ecuador. And I'm sure you've heard of Darwin and his famous finches, but they're all very well adapted to the environment they're living in, and they're all very different, although they probably evolved from a very similar ancestor. And they might have even evolved essentially from the same species, but over time, again, maybe they traveled, or maybe they were all one island at one point and then eventually got separated, or they formed new gametes, whatever it might be. Now they're reproductively isolated, and we've actually formed new species on each of these islands. Okay, and a very similar thing happens here in Hawaii. We have a lot of, like, say, fish that look the same and pretty much have all the exact same characteristics as some fish in the Indo-Pacific, but they are actually different now because they are reproductively isolated and they can no longer mate with one another, probably mostly because of location, right? They're in just such different locations, and for fish, let's say, to migrate across the ocean like that would require some really kind of freak event. Like you'd have to have some El Nino event where the water is a little bit extra warm, or some really crazy current that was happening. So it's not to say it's totally impossible, but it is very, very unlikely. And these are the kind of things that lead us into speciation or forming new species.